Welcome to Lesson 6 of Module 1, Cells as the Basis of Life. In this video, we're going to be looking specifically at enzymes and how they work to catalyze biochemical reactions within a cell. Again, we're looking at how cells coordinate activities within their internal environment and the external environment. Some of the meta-language we're going to use today is an enzyme, which is a special large protein. Optimum is the word we use to refer to the conditions under which enzymes perform at their best. A catalyst is a substance that accelerates the rate of any chemical reaction without themselves being changed. Denaturation is the loss of the characteristic 3D shape of a protein, which prevents functioning of the enzyme, and it is irreversible. The substrate is a molecule upon which an enzyme acts, and a cofactor is any extra ion or molecule that works with an enzyme. So, enzymes, being special large proteins, they in fact regulate organisms and maintain the rate of its chemical activity. So they have specificity. So different enzymes act as catalysts for different biochemical reactions. And they do so by binding, almost joining to a specific type of molecule called a substrate. They have catalytic power. So they don't actually make reactions occur. So the reaction would occur on its own. Instead, they make the reaction occur more quickly. And the good thing about an enzyme is that it can catalyze repeated reactions. So they're not consumed or altered in the reaction. So they do not form part of the products. They are the same as what they at the end as what they were at the beginning. Some characteristics of enzymes is that they're made of proteins. They're catalysts because they control the rate of chemical reactions. They remain unchanged. They're only needed in small amounts and they're highly specific. So only one enzyme catalyzes one type of reaction. They work best under optimum conditions, and this specifically such as temperature, pH, and substrate concentration, but they may need coenzymes to help in their functioning. Enzymes take or provide an active site for reactions to take place. The area in which the substrate binds to the enzyme is known as the active site. And once that enzyme binds with the substrate, they create what is called an enzyme substrate complex. The binding induces a temporary change in the shape of the enzyme, known as an induced fit. Once the chemical reaction occurs and the substrate is changed, the products are released and the enzyme returns to its original form. When the temperature, pH, and the concentration of the substrate are optimal, so at the right conditions, the enzyme activity is at its highest, the rate of reaction at its fastest, and it's operating at maximum efficiency. So here we have just a model that models the enzyme activity process. So we can see here that the substrate are two triangles that are joined together. It binds to the active site of the enzyme. Once it's bound there, the chemical reaction is able to occur, and in this case, it's the breaking of the bond between the two triangle compounds, or the two triangle elements, and then we see that the enzyme in fact releases the substrates whose now formed products, and the enzyme is able to go and catalyze a new reaction. There are two models of enzyme activity which can be referred to in texts and in questions. The first was what was known as the lock and key model. And in this model, the active site and the substrate fit together just like a key would fit inside a lock. The substrate is the same shape as the enzyme's active site, and we can see here that when we've got our substrate molecules, they can break up, they fit exactly into that enzyme. Once that reaction is catalyzed, it's able to go free. That enzyme is able to catalyze a new reaction. The other model is what we call the induced fit model, which we referred to a little bit earlier. And so the active site is more flexible and it changes shape to fit the substrate more tightly. Once it's bound, it fits there, this reaction is catalyzed, and then the products are released, and then the enzyme is again able to go and catalyze a new reaction. The only thing that is different here is that the active site actually changes once that substrate is bound to the enzyme. Here we can see a graph of a reaction, and we can see here that without an enzyme, a lot more energy is required for the reaction to occur. With an enzyme, we can see that the total energy required is much less. We do get to the same state at the end, so we still, both reactions still occur, the product is still formed. We can just see the amount of energy that is needed to initiate reaction is much less with an enzyme than without an enzyme. Referred to earlier about some factors that affect enzyme activity. So, enzymes work best at a particular level of acidity, so a specific pH, and any variation above or below that specific level reduces the rate of activity. If the pH is too extreme, it can denature the enzyme, which causes a permanent change in its structure, and therefore the enzyme can no longer catalyze the reaction. Temperatures are the same. It has a range in which it works best, which is 37 degrees in mammals, and the speed of the reaction decreases if it varies either below or above that temperature. And concentration, so an increase in substrate concentration will increase the rate of reaction until all the enzyme active sites are occupied. So if there are 10 enzymes available, 10 substrate molecules can undergo a reaction. If there are any more than 10, obviously some substrate will have to wait until there is a free enzyme for that reaction to occur. So therefore we say that the rate of the reaction is decreased in those situations. That is lesson 6 and the last lesson in module 1, as cells as a basis of life. Make sure you tune in for module 2.